Chapter 17, Gluconeogenesis. Let's look at the big picture. In Chapter 15, we learned that the glycolysis is just an arrow from glucose to pyruvate. Then where is the gluconeogenesis? Let's zoom in this orange circle. Inside the orange circle, there's two arrows point to different direction. Glycolysis is from glucose to pyruvate. The arrow is down. Gluconeogenesis is the arrow up. It's from pyruvate to glucose. Why does our body need to synthesize glucose? Fasting is a part of many cultures and religions, including those native Indian tribes. Fasting is believed to cleanse the body and soul and to foster spiritual awakening. Gluconeogenesis is an important metabolic pathway during time of fasting or starvation because it supplies glucose to the brain and red blood cells, tissues that depend on this vital fuel. This chapter includes 3 sections. Section 1, glucose can be synthesized from non-carbohydrate precursors. Section 2, we're going to talk about the gluconeogenesis is being regulated, just like any other pathway. However, the gluconeogenesis and the glycolysis are reciprocally regulated. And the third section is a metabolism in contact. It's a case study. Here are some facts about gluconeogenesis. When does it need it? Is during fasting or starvation when the glucose level is low. Why? Because glucose is such an important fuel, primary fuel for the brain and the only fuel for red blood cells. Where a major site is in the liver, although can occur in the kidney and some happen in the intestine. What to be used? The synthesis of glucose from non-carbohydrate precursors. Section 1. Glucose can be synthesized from non-carbohydrate precursors. So the gluconeogenic pathway converts pyruvate into glucose. And where pyruvate can come from? One is from the muscle contraction. In chapter 15, we learned that during the intense exercise, the pyruvate, when there's not enough oxygen, the pyruvate undergoes fermentation and form lactate. That the lactate causes your muscle sore when you do intense workout, and that's from the pyruvate. And that process can be reversed. Lactate can reverse back to pyruvate. And the other source is from muscle, protein. We all learned that we know it's a common sense that when you're starving, you lose muscle. Like the people in concentration camp, they're really skinny. They don't have muscles. The muscle will break down to amino acid. And it's not the amine in amino acid that can be synthesized to pyruvate, because pyruvate is carbohydrate. It's the carbon skeletons of some amino acid that can be converted to pyruvate. And the other source is fat. When you're starving, you lose fat, and the fat triglycerol, and not the fatty acid that can be converted to glucose. It's the backbone, the glycerol, that can be converted to pyruvate and used for glucose synthesis. Let's look at the big picture again. Amino acid can convert it to pyruvate. Fatty acid, fat, after cleave the fatty acid, the glycerol backbone can be converted to pyruvate. This is the pathway of gluconeogenesis. The distinctive reactions and enzymes of this pathway are shown in red. The other reactions are common to glycolysis. In glycolysis, we learned there are three regulatory site, control site. The first one is phosphoenolpyruvate, from phosphoenolpyruvate to pyruvate. That control site, when reversed in glycogenesis, there's two new enzymes involved. The other control site, from fructose 6-phosphate to fructose 1,6-biphosphate, and to reverse it in gluconeogenesis, there's one new enzyme. And the third control site, there's another new enzyme. The enzymes for gluconeogenesis are located in the cytoplasm, except for the pyruvate carboxylase. It's in mitochondria. And this glucose 6-phosphatase is then in membrane bound in the endoplasmic reticulum. The entry point for lactate, glycerol, and amino acid are shown in the chart here. For acetate, the entry point is pyruvate. Acetate change to pyruvate. Some amino acid converted to pyruvate, some are converted to oxaloacetate. For fat, is the glycerol enters from the dihydroxyacetone phosphate. Gluconeogenesis is not a complete reversal of glycolysis. The previous slides, we know that there's 4 new different enzymes facilitate the gluconeogenesis. And let's look at gluconeogenesis from the free energy point of view, from the thermodynamic point of view. This chart, this figure is glycolysis from chapter 15. And in this glycolysis, there are 3 control reactions, irreversible reactions that act as a control site. I mark it as A, B, and C. Let's look at A. Is a glucose turned to glucose 6-phosphate, and the free energy is minus 33. This reaction we see in the left, there's ATP. So we also learned in the past that if there's one reaction doesn't happen, you coupling with a reaction that is exergonic, then the reaction may happen. So for this glucose to glucose 6-phosphate, it's irreversible because it's coupling with ATP. If not coupling with ATP, then glucose 6-phosphate may just turn into glucose if there's no ATP in there to help it to move to the right side. Okay, the B is the same thing. There's ATP in there to facilitate the coupling this reaction to make it irreversible towards to the right. However, the number C, the C reaction, that one, it didn't need, it generated energy. That's why it produce ATP. So that one is slightly different than A and B. See, this A and B has ATP help the reaction, coupling with the reaction to move irreversibly to the right. But for the equation C, is actually spontaneous without coupling with ATP. Let's first look at the control site C in glycolysis. The phosphoenolpyruvate changed to pyruvate produce 1 ATP. It's an irreversible reaction. This is a reaction because it doesn't need to consume an ATP but produce ATP. You know that this reaction is moving forward to the pyruvate side spontaneously, irreversibly. In order to reverse it from pyruvate to phosphoenolpyruvate, you have to add more, coupling more ATP, like the high energy phosphate compound to make it reverse its reaction. That's what happens. That when the pyruvate change to phosphoenolpyruvate, not only the two new enzymes involved, you also need coupling two high energy phosphate compound, ATP, GTP, two of them, to make it move to the reverse direction. I hope that makes sense. Previous slides, we learned that the pyruvate converted to phosphoenolpyruvate can be happen if there's two high energy phosphate compound involving support that reaction. And the detailed reaction happen in two steps. The first step is in mitochondria. The pyruvate produced through glycolysis is in cytoplasm, and that pyruvate has to enter mitochondria. The 3-carbon pyruvate combined with 1 carbon dioxide consume 1 ATP, catalyzed by the enzyme pyruvate carboxylase, meaning adding 1 carboxyl function group, form a 4-carbon oxaloacetate. This 4-carbon oxaloacetate add another high energy compound, GTP, in cytoplasm, catalyzed by phosphoenolpyruvate carboxykinase, meaning remove 1 carboxyl function group and turn to this 3-carbon phosphoenolpyruvate and release 1 carbon dioxide. This is a figure describe the steps. Osteoacetate is shuttled into cytoplasm and converted into phosphoryl pyruvate. Let's look at the glycolysis control site B. In that site, the fructose 6-phosphate irreversibly changed to fructose 1,6-biphosphate. It is an irreversible reaction, but it has to coupling with ATP. So for the gluconeogenesis, when there's no ATP involved, and the reverse reaction actually happens. So the fructose 1,6-biphosphate can change to fructose 6-phosphate with a new enzyme fructose 1,6-biphosphatase, meaning remove one phosphate function group. In glycolysis, the control site I marked as A is glucose coupling with 1 ATP, changed to glucose 6-phosphate with hexokinase as enzyme. That reaction is irreversible. But if you remove the ATP, if the ATP is not present, in gluconeogenesis, the glucose 6-phosphate reverse change to glucose with the enzyme glucose 6-phosphatase, meaning remove one phosphate. And we can see that in the picture that the glucose 6-phosphates enter the ER lumen, and the glucose 6-phosphatase is an internal memory protein, also act as an enzyme that catalyzes the removal of phosphate from glucose 6-phosphate and free the glucose. And then the fructose is transported to cytoplasmic site. This is the overall reaction equation that 6 high transfer potential phosphoryl groups are spent in synthesizing glucose from pyruvate. So you have the 6 high transfer potential phosphoryl group are 4 ATP, 2 GTP, and also need 2 NADH, meaning 4 electrons. All that gave to the pyruvate to form the glucose. And that reaction happening with the free G is minus, because the reaction coupling 4 ATP, 2 GTP reactions. This is the bonus question for this chapter. You can find answer in the book. The gluconeogenesis and the glycolysis are reciprocally regulated. When one is active, the other one is inactive. It's determined by the glucose, blood level of glucose, and if ATP is needed. When glucose is abandoned, the glycolysis is a predominate. When glucose is low, then the gluconeogenesis is highly active. When ATP is required, because glycolysis produces ATP, so this pathway is predominant. And glucose is required, gluconeogenesis is favored. This figure shows the reciprocal regulation of gluconeogenesis and glycolysis in the liver. We can see the reciprocal relationship between these two pathways. The level of fructose 2,6-biphosphate is high in the fat state and low in starvation. The F26BP is a key signal molecule in the liver. It signals blood glucose level. It's a potent activator of PFK, inhibitor of F16BP. The balance between this is the domain structure of bifunctional enzyme. One side is a kinase domain, one side is phosphatase domain. One side adds a phosphate, one side is remove a phosphate. And which side is more active depends on the blood glucose level. And how blood glucose level control this bifunctional enzyme is through a hormone. When the blood glucose is low, the hormone glucagon is secreted, and the glucagon signal pathway leads to the phosphorylation of the bifunction enzyme, which inhibits the kinase and stimulates the phosphatase. This is the figure shows the control of the synthesis and degradation of fructose 2,6-biphosphate. A low blood glucose level, as signaled by glucagon, leads to the phosphorylation of bifunctional enzyme, and hence to a lower level of fructose 2,6-biphosphate, slowing glycolysis. High levels of fructose 6-phosphate accelerate the formation of fructose 2,6-biphosphate by facilitating the dephosphorylation of the bifunctional enzyme. This is another bonus question. Insulin normally inhibits gluconeogenesis. We learned that the glucose can trigger the insulin secretion, and insulin is supposed to inhibit gluconeogenesis. In type 2 diabetes, insulin fails to inhibit the gluconeogenesis, and that condition called insulin resistance. Just think about you have high glucose already, and the insulin is supposed to inhibit the gluconeogenesis to produce more glucose. And in type 2 diabetes, even though you already have enough glucose, and your body still produce more through gluconeogenesis. The good news is exercise and diet can enhance insulin sensitivity. Metabolism in contact. Precursors formed by muscle are used by other organs. The muscle and liver display inter-organ cooperation in a series of reactions called Cori cycle. In muscle, when you do intense exercise, the muscle contract. We learn from chapter 15 that the pyruvate can change to lactate through fermentation. And the lactate will transport to liver and convert it to pyruvate and convert back to glucose through gluconeogenesis. And that releases the burden of muscle to digest the lactate. That's why when you do intense exercise, your muscle feels sore caused by the lactate buildup, but then after a while it goes away because the liver would metabolite the lactate into pyruvate and further synthesize to glucose for future use. And that cycle called Cori cycle.